The very last minute to introduce our speaker for today. Luckily for all of you and for me, she's my good friend, Catherine Lilly, so it is not the hardest introduction I've ever done. Catherine is a professor of the biochemistry department at Cambridge in the UK, and she's a fellow in Jesus College there. She has been doing some absolutely fantastic work on mass spec and on localization within cells for the past 10 years or so. About 10 years before that, she was pushing the boundaries of quantitation in mass spec. I was lucky enough to spend a month in her laboratory learning how to use new techniques, to me, new. Her lab were expert at them and was hosted wonderfully by she and her students. The most fun thing for, I think, all of us is that Catherine has been a member of the ABRF for 20 years now. She started out in what I consider the dark side, but the DNA people considered the light side, in the DSRG and the NRG committees, and then moved over to the protein side when she took a position at Cambridge. So she has been a longtime member, and I'm very much looking forward to her talk. Please join me in welcoming her.
I failed at the first fence. Okay, thank you very much, Laurie. Thank you for stepping in at the last minute to introduce me. So it's a great pleasure and honour to be here to talk to you today. As Laurie said, I came to my first ABRF meeting 20 years ago. It was in San Diego. And of all the meetings I've been to since, this had the most profound effect on me as a scientist. Not only because I was diagnosed with chickenpox varicella halfway through, and I thought I'd never come back to this flea-ridden country again, but I have, and have worked within the ABRF in many different guises for many years, and have felt very supported by the ABRF as I've transitioned from just only having a core facility within my laboratory to also operating a research group alongside the core facility, which we find to be very successful because one feeds the other. And actually, what I'm going to talk to you about today came via the core facility, the need to develop methods for which there was nothing off the peg that we could use to answer some pretty basic questions in cell biology. So what I want to talk to you about today is spatial proteomics and why proteins end up where they do in cells. So we all know that proteins have to get to the right part of the cell. They live in discrete subcellular niches where they carry out their function in a context-specific manner. They have to do this. They've got to get to the right place. They've got to find their binding partners. If they're enzymes, they have to find their substrates, and so on and so forth. And how they get there is still a bit of a mystery. It's very dependent on localization signals, on interacting partners. It can be influenced by the post-transcriptional status of the protein, so it may be a result of alternative splicing. It can be a result of post-translational modification, the structure of the protein, and even where the protein is synthesized, so where it's translated in the cell. And why we're interested in this is because there are plenty of examples of disease which are caused by this process going wrong. So a protein, if it ends up in the wrong part of the cell, can lead to some pretty nasty disorders through mutation, through aberrant protein modifications, aberrant interactions, aberrant protein folding, and aberrant translation localization. So if a protein's made in the wrong place, then that can give rise to some pretty horrendous disease states. So what my research lab is all about is to dissect the relationship between transcription, translation, and trafficking. So we know where RNA is synthesized, it's transcribed in the nucleus, but then it has to get out and get itself translated. And it's really the order in which this happens. So does translation happen and then the protein product trafficked to the right place? Or is the RNA trafficked and it's then translated in the correct location? Now, in the case of proteins that are trafficked through the secretory pathway or who are membrane proteins, we know that the RNA ultimately is translated at the endoplasmic reticulum. But for soluble proteins, we're not entirely sure where those transcripts that give rise to these proteins are translated, and if any of it is translation in situ. So to be able to answer these questions, we need to ask some pretty fundamental questions and develop technologies to be able to answer them. So can we achieve a map of the proteome on a global scale with high resolution? So can we get a map, a sort of a street map of the cell? What is the impact of post-translational and post-transcriptional translation on the location of a protein? What changes happen upon perturbation? So how do proteins move around? Do proteins adopt different structures and functions in different parts of the cell? And this gives light to a concept of protein moonlighting. So proteins may have different jobs in different parts of the cell, which would make some sort of sense because actually our proteome is really quite small for the number of functions that have to go on inside a cell. Where are proteins translated? Why does this happen and how does it happen? And what's the role of RNA binding proteins in this process? So RNA is not let out on its own. It's a very fragile molecule. It's often coordinated by proteins. So the whole of this trafficking process, what is the role of proteins in this process? What's the role of untranslated RNA and what's its subcellular localization? And are there intracellular pools of proteins, or different intracellular pools? And if so, and you've got these proteins in different parts of the cell doing different jobs, are those pools interchangeable or are they discrete? So are they produced in different pockets, different subcellular niches, and the two different pools of the same protein never interchange? So to be able to answer these questions, we need to be able to use and develop some methodologies which will allow us to probe further the relationship of the proteome in 3 dimensions and its corresponding transcriptome. So we need to develop methods which will give us a comprehensive map of the whole cell in a single experiment if we can get away with it. The protocol needs to be able to maintain RNA integrity, so we can fish out RNA from the same subcellular sampling as we do the proteins. We must be able to extract the RNA and RNA binding proteins, and we must be able to use all these methods dynamically. So these are quite bold aims. We've not got quite to the end of this. We've not produced all the methods and certainly not road tested all the methods that we need to answer these questions. So what I'm going to give you is details of some methods that we've produced, how we've used them, but also go on and talk about a lot of unpublished work as well. So if you want to know where a protein is in a cell, you have a variety of different choices of how you can go about working out the protein's address. If you know what you're interested in, then you can use microscopy methods. You could use a fluorescent antibody or GFP tag your protein, and then use microscopy to see where the protein is. So that's great, but it's not particularly high throughput. More high throughput methods perhaps would answer a slightly different question, which is where you have a subcellular niche that you're interested in. Let's imagine that you're all fascinated by the mitochondria. And so what you can do is to purify the mitochondria as best you can from a cell and then get its parts list. So use very traditional mass spectrometry methods to identify the proteins that are there. Other very cool methods to do more or less the same thing would be to use proximity tagging. So here you would direct your ascorbate peroxidase or an enzyme which, when it's in situ, will biotinylate all its neighbors. And then you fish out any protein that's biotinylated, and that will give you information about what proteins are present in your subcellular niche of interest. Now, these are very powerful methods, have been used to great effect, but they only give you one piece of information, and that is, my subcellular niche contains these proteins. What it's not telling you is if these proteins are present in more than one location. So what we've been working on now for many years, for 14 years or more, are methods that don't require you to purify any subcellular niche to homogeneity, because most subcellular niches cannot be purified, but allow you on a cell-wide basis to look at distributions of proteins through subcellular fractionation, so through biochemical fractionation. So these methods are really based on work done in the 1950s by this guy here. So this is Christian de Duve. He died a few years ago. And he used a method called correlation profiling to look at where proteins are in a cell. And by applying these methods, he discovered both the peroxisome and the lysosome. So the idea is as follows. You don't try and purify anything. You use some form of subcellular fractionation, mostly but not exclusively centered around different methods of centrifugation. And then you look at the abundance of proteins from known subcellular niches throughout your fractions, and anything which has got a similar distribution, you would then say is from the same subcellular niche. So, for example, if we were to carry out density centrifugation, we'll take some fractions, and then we'll have a look at the abundance of different proteins in our fraction. Let's imagine these green lines here are the distribution, the correlation profiles of Golgi proteins, so proteins from the Golgi apparatus, and anything showing the same distribution you would assume is a novel Golgi protein. And the premise here is that each subcellular location, so each organelle, will give you a slightly different correlation profile, and so you can then match the unknown correlation profiles to those organelles. So the method that we developed a while ago based on de Duve's principles we called LOPIT, or the localization of organelle proteins using isotope tagging. And the way that we did this was to gently lyse our cells using a ball-bearing homogenizer. We then used density centrifugation such that an organelle will migrate down the tube to a point during centrifugation which is consistent with its buoyant density. So very heavy organelles will end up somewhere near the bottom, the lighter ones somewhere near the top. We take fractions. Our method has us using isobaric tagging, so we use TMT, so that each fraction would be labeled, or the peptides derived from it would be labeled with a different TMT tag. And then we use tandem mass spectrometry. And this time, our mass spectrum, our tandem mass spectrum, has got nice fragment ions from which we can deduce the sequence of the peptide and therefore the identity of the protein that it came from. But importantly, our low mass reporter ions, which fall off each tag in a sample-specific way, give us the distribution of that peptide and therefore the protein it came from down our gradient. So this particular peptide would have come from a protein where there was more of it in this fraction, so this particular peptide would be associated with a protein from a lighter organelle. We have a huge amount of data, so we have to use some fancy informatics methods, and I'll talk about those in a minute, to be able to visualize our data and our correlation profiles. The easiest way to do this is to use principal component analysis, where proteins that have got similar correlation profiles will cluster together. So you can see here, anything in red are markers for the endoplasmic reticulum, and so the grey dots, which are not assigned, you would then use some form of machine learning method to assign proteins with very similar correlation profiles to that organelle. So we can do this on a cell-wide scale, although in this particular example, which is very old, the eagle-eyed members of the audience will notice that we've got no nucleus. So early on, we weren't able to capture the entire cell, but only a subset of organelles of interest. So what this method shows us is the steady-state distribution of proteins, and proteins that don't assign to any of these clusters we call no man's land, which is not necessarily the best word for it, but that's the one we came up with years ago. Doesn't go down so well in certain audiences, particularly those that think that we should call it no person's land, because we're being a bit sexist. So why a protein would end up in no person's land is because either it is part of a cluster which we have no markers for, so we can't say it's a particular organelle, or that the protein is present in more than one location. And if that is the case, then the correlation profile will be a mixture of whichever locations the protein can reside in. So as time's gone on, we've made this method better, we've optimized it, and a couple of years ago we produced a new version of the method, which we call HyperLOPIT. The idea here is to try and get complete coverage, so we don't throw anything away. So we do a separate nuclear enrichment, and then after lysing our cells, we make membranes, so this is all the organelles. Anything which doesn't cushion onto our gradient is enriched in cytosol, so that's another fraction. And then the membranous organelles are separated again using density centrifugation. So now we've got a nuclear-rich fraction, a cytosolic-rich fraction, and the rest of the organelles by the fractionation of our centrifuge tube. We'd use a Western blot to make sure that we've not smashed the cells to smithereens, and we do have some organelle-specific densities in our fractions, and then using the power of multiplexing with TMT and methodologies developed on thermo instruments, so we do most of our work on the Orbitrap Fusion Lumos, so that we can do MS3 to improve the accuracy of our TMT quantitation. We then follow this up with some pretty nifty machine learning tools to be able to classify our proteins and assign them to different subcellular locations. So the method is in 4 bits, fractionation, multiplexing with our isobaric tags, LCMS, and then machine learning, which allows us to interrogate our data. So this is the sort of plot that you would get. Again, we're looking at this in principal component space. So this is PC1 versus PC2. We've got 10-dimensional data, so this is just looking at the first 2 dimensions. And we have very nice separation of our organelles. We've also got sub-organelle clustering. So here are proteins that are associated with the nucleoplasm. Here are proteins which are enriched in chromatin. We have the mitochondria floating above it, and another principal component, you'll have to believe me here, is the peroxisome. And we have the secretory pathway all the way from the ER right out to the plasma membrane. We have a nice cluster associated with the cytosol, and also, for free, large protein complexes. So in pale pink is the proteasome, and these two here in the center of the plot are the different ribosomal subunits. We've also got information on recycling vesicles too. Important to note is that half the proteins do not classify. So we're saying that half the proteins of the cell are in more than one place. So what else can we do? Because we're looking at 8,000 to 9,000 proteins per experiment now, we've got sufficient coverage to look at things holistically. So we mapped the major signaling pathways, and it's nice to see that they start in the plasma membrane, at the receptors, all the way through to the terminal transcription factors associated with the chromatin. We can look at protein isoforms. This particular protein, leucine aminopeptidase 3, has got 2 forms, one in the mitochondria and one spread in an intermediate position between the nucleus and also the cytosol. And we can also look at protein complex components. We don't do anything to keep them together, but they still cluster very nicely with one another. As I said, we have had to produce a lot of different informatics solutions to be able to interrogate our data thoroughly. So we have methods where what we do is to use other types of data, other forms of data. And this is a process called transfer learning, where we take information about the proteins. It could be their sequence, it could be from imaging, it could be who they interact with. And we use this information to add certainty to our classification of proteins to novel subcellular niches. And we've also recently produced some nice R-based tools to be able to optimize or help optimize experiments to see if you start to change the shape of gradients or the different centrifugation times, how your clusters are resolving from one another. So when we first started to publish our LOPIT data, the reviewers said, oh, well, that's all very nice, but you need to go off and prove it. You found all these novel proteins from different parts of the cell. We want you to go off and do some confocal microscopy to prove that your novel plasma membrane protein actually is in the plasma membrane. Now, we weren't in a position to do this. We don't have the resources in my lab to do this. So we teamed up with the Human Protein Atlas people in Stockholm. So this is Emma Lundberg, Peter Tull in her lab, and the big boss, Matthias Ulen. And I said to Emma, if we were going to compare our data or our method with what you're doing, what would be the best cell line? So Emma and her crew, what they're doing is to produce as many fluorescently tagged antibodies to human proteins as they possibly can, and they are amassing vast amounts of confocal images for the human proteome. So she suggested that we used U2OS, which is a human osteosarcoma cell line. So Katerina Galadaki, who's a PhD student in the lab, went off and got a very nice HyperLOPIT plot, and here it is. Yet again, half our proteins are in more than one place. And we challenged our localizations against what Emma and her team had got. So these are completely orthogonal methods, and there's no reason why these methods should give similar data. They are keeping their cells intact. They've got single cell information. What we're doing is to use a ball-bearing homogenizer to break up our cells, and it could be that our proteins are floating off and ending up associated with all sorts of different subcellular niches, which is an artifact of the way, or could be an artifact of the way that we carry out our experiments. So we compared with their thousands and thousands of immunocytochemistry images. And firstly, what they had found, like what we had found, is that half the proteins are in multiple places. So that gave me a very warm, fuzzy feeling that if they found it with their method, then the fact that our proteins are also in multiple places isn't a manifestation of the way that we're carrying out the lysis of the cell, all the centrifugation steps that we use. So what we found, to our surprise and great delight, was that the two data sets actually had much in common. And the agreement for certain subcellular localization, such as the endoplasmic reticulum and the mitochondria, was very high indeed. There were some differences between the two methods, but interestingly, our agreement scaled with their antibody reliability. So they know how good their antibodies are, and the ones that they feel are their sort of gold antibodies, there was a very, very good agreement between the two. And we published this together with them, or as part of their main paper about their cell atlas last year in Science. So the HyperLOPIT method is not for the faint-hearted. You often, depending on how your cells grow, have to stay up all night. PhD students don't like it so much. So we thought that we would try and come out with simplified methods to enable higher throughput and also methods that wouldn't take as long or were expensive on cells. So we now have three methods. Only one, the HyperLOPIT method, is yet to be published. In the lab, we call them the Rolls-Royce method, the Toyota Corolla method, and the skateboard method. So the skateboard method it's also known as lazy LOPIT. So one gives maximum resolution, that's the Rolls-Royce. One is for when the sample is limited, and here, rather than using density centrifugation, we're using differential centrifugation. And then we have our skateboard method, which is quick and nasty, but its main reason for being is to maintain RNA integrity. So this is a method that we want to use to not only look where proteins are in the cell, but also to look on a global scale where their corresponding transcripts are. So these are the differences between the two, the three rather, in terms of the number of cells that are required and the resources, including mass spectrometry time. And to compare the Rolls-Royce and the Toyota Corolla, we have this slide. The first steps are the same for both. We use a ball-bearing homogenizer. But the main difference is that one relies on density centrifugation and one relies on successive differential centrifugation each time we're spinning out a different set of organelles. Once we have done this separation, then the mass spectrometry and the machine learning is the same for both approaches. So this is unpublished data, but it gives you an idea here of the difference in resolution between the two methods. So you can see here on the left-hand side, this is with U2OS cells as well. This is our LOPIT DC, so this is the middle method. This is the resolution we get, and you can see that there is better resolution using the HyperLOPIT method, but actually the resolution that we're getting from the LOPIT DC method is sufficient for most cases. And you can use all sorts of methodologies to see how good your methods are by taking a subset of the data out during the analysis and putting it back in again and seeing how successful when you reanalyze your test data set against the training data set that the reclassification is. And the methods are certainly comparable when it comes to looking at proteins and different organelles, looking at signaling pathways, looking at various complexes. Importantly, if you start looking at our data and looking at pathways that are pretty fundamental metabolic pathways, the data sets are so rich that whatever you really probe the data with, there's always a few surprises. And I just want to draw your attention to this surprise, because it hopefully will come back as with an answer to what could be going on towards the end of the talk. So this is glycolysis and gluconeogenesis. The textbooks tell us this takes place in the cytosol and the mitochondria. And what we found was this is perfectly true, but in the case of some proteins, they're in multiple places. And each time, it's the proteins that are responsible for the flux through the pathways. So the kinases, hexokinase, phosphofructokinase, pyruvate kinase, that control flux through glycolysis, those proteins are in more than one place. We have a GUI, so if you've got a favorite protein, you can bring up our website, type in your favorite protein, and it will show you on our maps in a few different cell types now where it is in the cell. So just to show you that we can use it for something other than human cell lines, this is a project that I have with an Australian colleague of mine in Cambridge, Ross Waller, and his Russian postdoc, who's now just about to become an independent fellow. And what they were interested in doing was to find subcellular location of proteins in a parasite. And the parasite that they're interested in is Toxoplasma. This is the one that all your cats have got, and if you have a cat and stroke it too much, you've got it too. So cats are the definitive host. Any warm-blooded animal is the intermediate host. We might have it, but we wouldn't know we'd got it unless we become immunocompromised, and then it can be fatal. So it's a parasite, it's part of the Apicomplexar family, and it's got some very specialized organelles. And so far, 50% of proteins that are thought to be produced by Toxoplasma have no known location and no known function. So this is a beautiful map got by Konstantin. He's used a slightly different way of portraying the data. This is a t-SNE plot, but it's the same sort of idea as PCA. And he's got the most gorgeous resolution. So he can now map of these 50% of proteins for which we have no idea what they do in Toxoplasma, he can now map them to to the subcellular locations, the different organelles, including the parasite-specific organelles. And when we look to see, compared with a CRISPR screen, where a group, I think somewhere in the States, knocked out systematically these proteins, what we find is that the proteins that are involved in infection of human fibroblasts are actually partitioning to the same subcellular niches. So because we are now finding that there are novel proteins within these niches, we can start to use them as therapeutic targets. So this is a very powerful technique. I said to begin with that we have to work out whether, you have to be able to use our methods dynamically. So we want to see what happens to proteins when we do something to the cell. How do they move around? So just to show you that the LOPIT method can cope very well with perturbation and relocalization, Here's a project that I started a few years ago now with Andrew Smith from UCL and a former postdoc in the lab, Claire Mulvey. And what they're interested in is what happens when you stimulate the innate immune response. So they challenged THP1 cells, which is a monocytic-like cell line, with lipopolysaccharide, which triggers this innate immune response, with the idea that they wanted to see if we could see that proteins were relocalizing. So they carried out replicate parallel experiments. On the left-hand side of this plot is the LOPIT plot of the cells before stimulation with LPS, and on the right-hand side, what we find when we stimulate with LPS. So we've got these two maps, and now we can play compare and contrast. And there are two things that we have to see. So we want to look at proteins that are not present when there is no LPS, but they're expressed upon treatment with LPS. So the whole load of interleukins that appeared in locations that you would expect. We also wanted to look at the reproducibility, so sets of proteins that we wouldn't expect it to have moved. But then, and this is still work in progress, we got some very nice translocations of proteins, protein kinase C-alpha from the cytosol to the plasma membrane, where it finds its substrates upon stimulation, and also transcription factors, particularly STAT6, which migrates from the cytosol into the nucleus upon stimulation. So this method can be used dynamically. So that sort of concludes the protein bit, or the whole protein map bit. But I said that we're interested in RNA and where that is. So what we had to be able to do was to fish out RNA coordinated with proteins, then ultimately RNA and RNA binding proteins from our different subcellular fractions to give us an overall picture of the relationship between the proteome and the transcriptome. So we've had to develop a new method to capture RNA binding proteins, and I'll tell you why it had to be a new method in a moment. But this is work of a team of researchers that are employed on a Wellcome Trust Investigator grant to myself and Anne Willis, who's a very old friend at the University of Leicester. And the work I'm going to describe is mostly the work of the three postdocs on the grant, Rainer Queiroz, who's a mass spectrometrist, Tom Smith, who's an informatician, and Eneko Villanueva, who's an RNA biologist. So if you want to fish out RNA and the proteins that are bound to it, the most typical way to do this is to cross-link the RNA to the proteins, and then to fish out the RNA based on the fact that it's got a poly-A tail. So if you use beads which are derivatized with oligo-DT, you can pull out the RNA and anything that's bound to it. Now, there is an issue here. In fact, there are two issues. One is your RNA has got to have a poly-A tail, which not all RNA species has. And also, this is a very inefficient method, and you need an awful lot of cells to start with. There have been very recent papers, in fact this year, where to get round this, what the researchers have done is to come up with two different methods, two different groups. One's method's called CARIC, one method's called RIC. They do more or less the same thing, which is to grow the cells in the presence of 5-ethanoidyluridine, EU, and that gets incorporated into the RNA. And by click chemistry, you can then biotinylate the RNA and pull it out with streptavidin. So this is more sensitive. It doesn't require you to have a poly-A tail, but it does require you to grow your cells in the presence of EU, which they don't like very much. So it's limited to some extent in the applications for which it can be used. So what we did was to come up with a method that reduced this many plates of cells, which is what are typically required for using the oligo-DT method, to only needing half a plate of cells, and it's more sensitive, I think, also than the CARIC and RIC methods. And it is remarkably simple. It's so simple we had to give it a stupid name. LOPIT isn't a clever name, but OOPS is an even more stupid name. We did think that we'd change it for a while, but it's stuck. So it stands for the orthogonal organic phase separation. So the first part of it's the same. You UV treat your cells, so the RNA binds to the protein, and then you use organic aqueous partitioning, and you fish out any RNA, in fact all RNA molecules, more or less, or of a certain size, and all the proteins that are bound to them. And this is how simple it is. you just use trisol. You use a trisol extraction. Now, my understanding of trisol is that the RNA goes into the aqueous phase and the protein goes into the organic phase, and the methods that explain the protocol, how you would use trisol to extract RNA, say whatever you do, don't touch the interface. But it's the interface which we're interested in. So the interface is where our RNA binding proteins are. The RNA part of the cross-linked RNA protein complexes tries to drive the complex into the aqueous. The protein tries to drive them into the organic, and they end up stuck at the interface. We can then take these interfaces, we then do 2 replicates, and then we've got a choice. If we're interested in the RNA, we treat the interface with proteinase K. If we're interested in the protein, then we treat the interface with RNA. So I will give you some proof that this works very nicely. So if we're interested in the protein-bound RNA, we treat the interface with proteinase K and then repeat the trisol extraction, and now our free RNA ends up at the aqueous phase. To cut a very long story short, we proved that the RNA that we're extracting is what we think it ought to be, and what we're extracting all RNA more than 200 bases long. Less than that, there is less chance of the protein being coordinated with protein, and so it's not going to end up at the interface. We did next-generation sequencing to see what we got, and we also carried out multiple replicates and proved that this method is eye-wateringly reproducible. We were able also to find out another piece of information by looking at where the reads were along our RNA length when we compared a situation where we cross-linked RNA to protein and we hadn't cross-linked. So the case I'm showing you here is actin, and we've got 2 sets of reads where we haven't cross-linked and 2 sets of reads where we have cross-linked. And you can see that the read coverage, the read density, has gone down significantly in this area, which is an exon-exon boundary, and this area here, which is the 3' UTR of this particular gene, which gives us evidence, and we've got many replicates to show this, that this is the site in this particular RNA where protein is being coordinated. So that's it for the RNA. How's the protein doing? So in this experiment, we wanted to show that protein is being held at our interface by RNA and not for some other reason. So we carried out a SciLac experiment where we took the interface from cells that were heavy labeled. So the proteins would have all been heavier, they have heavy lysine and arginine in them. And then we did a replicate experiment where we treated the interface and therefore released proteins into the organic phase. We took those proteins, merged them with the proteins from this interface, and then looked at the ratios. So if proteins are being released from the interface by RNAs, we would expect the ratio to be 1:1. If the proteins were hanging around the interface for some other reason, and it wasn't because of RNA, and they were still then present in this experiment, we would expect the ratio to be less than 1. So having done that, what we proved was that actually the majority of the protein is at the interface because it is being coordinated with RNA, and if you take the interface after you've treated with RNAs and run it on a gel, you get pretty much nothing. So therefore, we're very confident that we can recover all the RNA, and that the protein, which we're also recovering, is at this interface because it is an RNA binding protein. So this is the underlying premise of the OOPS method. We can do all sorts of analysis on the proteins that we then subsequently identify by mass spectrometry. We can show that the method is very reproducible, and also that we are fishing out additional RNA binding proteins that are not found either by the oligo-DT method or, in fact, these click chemistry methods that I just described. We can look at the molecular function and the biological processes by looking at the Go ontologies of the proteins that we pull out. And it's interesting to see what new ontologies are appearing in our novel RNA binding proteins. So there's clearly a lot of proteins in cells involved in all sorts of different functions, which are RNA binding proteins. So this is opening up a whole new area and questions to why these proteins are finding it necessary to bind RNA. So to try and work out where the proteins are binding RNA, we carry out now an experiment to work out the sites of RNA binding. So imagine that this is our protein and RNA hanging off of it. So this is the area of crosslink. We use a protease which gives us quite large portions of the protein. And then we'll use a second protease with less specificity to really chop down still further to get smaller peptides. And now we've got two sets of peptides. We've got the peptides which are proximal, so these are next to the area of the protein which is binding RNA, and we've also got the peptide itself with RNA hanging off of it. If we use RNAs at this point, then that's going to cut the RNA molecule back down to one or two different bases, and then we can use mass spectrometry to work out the sort of domain information of where the RNA is binding, but more specifically, the residue which is cross-linked to the RNA. And to be able to do this is quite difficult because it's difficult to exactly predict what the base will be, which is left behind after RNase treatment. And so we have to use quite open mass spectrometry searches. I've been using a new tool developed by Leonard Martins and his group in Ghent. And we've got evidence now for many different RNA cross-link sites in both the proteins that we know are card-carrying RNA binding proteins, because the databases tell us so, but also the novel proteins. And this is still very much work in progress. And what we've been able to do is to look at the RNA binding protein now through evolution, all the way from E. coli, which of course is going to have RNA with no poly-A tails, and so is the RNA binding proteome of bacteria generally have not been that well studied, all the way through yeast and plants and different human and mouse cell lines. So when we look at the RNA binding proteins, or the number of different sorts of domains that we find in our cells, or in our proteins rather, we see that within our novel set of RNA binding proteins, there are some non-canonical domains that are appearing. So it seems that RNA is able to coordinate with different proteins in a way which has not been fully explored to date. So because we can start with a very small amount of cells, we can now use this in a dynamic application. And what Eneko Villanueva in the lab decided to do was just to follow the RNA binding proteome and how it changes through the cell cycle. So taking our U2OS cells, we treated with nocadazole so that we'd got cells that were trapped in M phase, and then they were and the cells that were enriched in the other parts of the cell cycle as well. And we needed to do two different things. We wanted to know how the total protein was changing throughout the cell cycle, and then how much of it was, or what the change in coordination of the RNA binding proteins. So it's important to see if an RNA binding protein is going up and down in the way that we measure its expression. Is that because the total amount of protein is changing, or the occupancy, so the amount of that protein which is binding RNA, is that changing? So we carried out our experiment. Here's the heat map for the total proteome. There are lots of different studies in the literature looking at how the proteome changes during the cell cycle. So we were able to make sure by looking at other data that the results that we were getting made sense. And then what we did was to carry out the experiment, this time pulling out the RNA binding proteins using the OOPS method that I've described. And to then have a look at proteins that were changing in abundance and proteins that were changing in their ability to bind RNA. And what we saw, that there was interesting sets of proteins that seemed to be increased in their RNA binding capacity as the cells were progressing from M phase to G1. And one of the interesting sets were proteins, again, going back to where I was probably 3/4 of an hour ago now, involved with glycolysis and gluconeogenesis. So what we found for the glycolytic enzymes, for example, is that they are RNA binding proteins, And although their absolute amount doesn't change throughout the cell cycle, their RNA occupancy does. So they seem to bind RNA, they get to M phase, they let go, G1, they bind it again. So why this is, and this is true for almost all of the glycolytic enzymes, the exception being aldolase. So the reason that they bind RNA and the reason that they let go in M phase is open to suggestion. So going back to subcellular location, where are our RNA binding proteins? So here's our U2OS map, and these were the RNA binding proteins that we knew existed before we developed OOPS. And there's a lot in the mitochondria, there's some in the cytosol, there's an awful lot associated with the nucleus and not much else. Now when we add the proteins, the additional novel RNA binding proteins that we found using our OOPS method, we've got a lot more of them. A lot more of them are associated with the cytosol. There's an awful lot of them in no man's land, or the proteins are in no man's land. That's their steady state location is more than one place. And intriguingly, a lot more associated with the secretory pathway, including membrane proteins. So I said our ultimate goal is to be able to capture the RNA in our subcellular fractions and not only work out what proteins it's bound to, but where that RNA is in the cell. So this brings me back to lazy LOPIT or skateboard LOPIT or whatever you want to call it. And this has been a real labor of love by Eneko Villanueva because we had to completely revamp the methods. We had to fine-tune the cation concentrations that we were using to stabilize RNA, to stabilize ribosomes, and particularly those that are associated with the ER. And the method also had to be, we had to be able to cross-link the cells first because we want to cross-link the RNA to the proteins. But this has all worked very nicely indeed. These are fairly new results. It's still work in progress. But here's the QLOPIT plot of cross-linked cells. So this is where the proteins are. And although we haven't, in the same fractions, yet done next-generation sequencing, what we have done throughout our fractions from our gradient is to carry out qPCR on transcripts that are in known subcellular niches. And what we found is that despite everything that we've done to these cells, that the method is likely to work because we've got very, very nice and promising clustering of RNA species that are known to be in the same subcellular location. So in green down here, we've got species that are mitochondrial. In blue, species that are associated with the ER. We've got the exist, the GIST RNA, which is nuclear, and some nice cytoplasmic or cytosolic RNAs. So we think now the world's our oyster. If we can get the correlation profiles using qPCR on a few transcripts, then next generation sequencing should be able to give us the correlation profiles of the whole transcriptome through our gradient, and so that hopefully we will, next time I talk to any of you, we'll have a map that I'm showing here the protein map, we'll have a corresponding RNA map and then can map the two together to see how the steady state location of a protein corresponds to the steady state location of its transcript. So in conclusion, I hope I've shown you that mapping subcellular location can give insight into the functions of proteins. I hope I've persuaded you that half the proteome lives in more than one place, and that if you combine different methodologies, you can actually get exquisite resolution on a global scale of where a protein is in the cell. I've also shown you new flexible pipelines to look at the spatial transcriptome, and really how using these methods are throwing up some very unexpected observations about the location of particularly proteins, and I'm sure RNA in cells. So all that's left for me to do is to acknowledge the great number of people over the years who have collaborated with us, particularly Anne's group in Leicester. The people in my group, if they are in red, they're informaticians. If they're in blue, they're not. If they're in italics, they're PhD students. The core facility I've also acknowledged because they have been extraordinarily helpful and supportive during all of this technology development. And here we all are last November, looking a bit cold, stood outside my department in Cambridge. So thank you very much for your attention, and I'm very happy to answer any questions.
What about the membrane proteins?
Pardon?
Membrane protein.
We see they're there. They're in all our plots. So we identify membrane proteins in the same way that we identify soluble proteins. So our classification to the different organelles, if they're membrane proteins, if they were a mitochondrial membrane protein, then they classify in the same way as the soluble proteins do to the mitochondria.
Lovely work. I am interested in the proteins that are in the act of being translated.
Mm-hmm.
Do you separate those would also get cross-linked, would they not? Is there any way you're separating them out or any way that you can identify them?
There are other mechanisms or methods that I would suggest for looking at actively translated proteins. I think in the terms of the proteins, you assume that the majority, the steady state of a protein is have already been translated and has moved to the right part of the cell. Whether that is also true for RNA, I don't know. But I think what our method will show, if the steady state location of an RNA molecule is that it is being translated or that it is being stored in a particular granule somewhere, then that's what we will see. We also plan to combine our methodologies with ribosomal profiling. So if you can fish out the ribosomes at different parts of the cell and work out what it is that they are translating at that part of the cell, then that's going to be, I suppose in many ways, the missing link in the process.
It'll be fun.
Any more questions? Okay, thank you.